Hi everybody, so I thought I would go over this experiment with you since I was not here today, and hopefully this will kind of help understand where we got our answers because it was kind of a tricky activity. So I'm first going to start by reading the experiment and what they were trying to tell you from here. So nucleic acids were known for 75 years before their importance in living cells was fully realized. The mystery of how DNA acts was first explored in bacteria. Research biologists first focused their attention on the role of DNA in a particular species of bacterium, Diplococcus pneumoniae. As you might guess from its name, this is the type of bacteria that causes the disease pneumonia. The name of the genus Diplococcus refers to the fact that the bacteria cells are in pairs and are found in round in shape. So diplo is double and coccus is berry shape. A shorter, more common name of the species is Pneumococcus is often used. There are actually two main kinds of Pneumococcus bacteria. One kind, each pair of bacteria cells is surrounded by a capsule of a sugar-like substance. The other kind has no capsule around the cells. Both kinds can reproduce, giving rise to cells like themselves. When cells with capsules divide, they form new cells with capsules, so they reproduce with the exact same as the parents. Cells without capsules form new cells without capsules. The capsules around the Pneumococcus cells is related to another characteristic of these cells. The cells with capsules cause pneumonia and subsequent death when they are injected into mice. Pneumococcus cells without capsules do not cause pneumonia in the mice that are injected. Now, I told you this a little bit on Friday, but this would be really cool to actually do this experiment, but because we're dealing with mice and Pneumococcus is a dangerous bacteria, we're not able to perform it. So I still wanted you guys to experience it, and that's why we're reading through this together in class. So let's take a look at the experiments. Experiment 1. We had healthy mice and they were injected with a few Pneumococcus cells without capsules, and the mice remained healthy. As remember we said the Pneumococcus cells without capsules do not cause pneumonia. In experiment 2, we did the same thing, only we injected them with Pneumococcus cells with capsules. The mice died, and the autopsies showed that the death was caused by Pneumococcus. Experiment 3, we did Pneumococcus cells with capsules, and we actually killed the capsules with heat, so the capsules were killed by heat. These dead cells with capsules were injected to healthy mice, and they remain healthy. In experiment 4, we injected the mice with a mixture of living Pneumococcus cells without capsules and a few heat-killed cells with capsules, so kind of a mixture there. Question 1, based on the results of experiment 1, 2, and 3, what do you think happened in experiment 4? Now, some of you might have skipped ahead and looked at the picture, but if you didn't look, what do you think happened to the mice in experiment 4? Well, a lot of you probably thought that the mice would live, because the strain, the rough strain, or the one with capsules was killed by heat. So because we didn't have the capsules, it should have killed the mice. So a lot of you probably thought that the mice actually died in experiment 4. Let's actually take a look here. Now, I wasn't here Friday, so I feel like a lot of people are probably confused by the rough and smooth, and what did that mean? Well, the rough strain is going to be the one, and you can actually see here, it's going to be the one without capsules, and that's what causes the mice or the mouse to live, because if it doesn't have capsules, it doesn't cause pneumonia. Smooth strain is going to be the one, you can see it here, it actually has capsules around it, and that's what gives the mouse pneumonia. The third one, we had heat-killed smooth strain, and it allowed the mouse to live because the heat killed the capsules around, causing it not to give pneumonia. Now, here's where it gets a little tricky. So we have some smooth strain, we have rough strain that was killed by heat, and somehow it still killed the mouse. So what I wanted to know is what kind of hypothesis you guys came up with. Now, I'm going to tell you a few students I got from the past, and let's see if you guys came up with some. So the first one, you could have maybe thought that the heat-killed smooth strain wasn't heated to hot enough temperatures. So some of the strain was actually still alive. So maybe some of you guys put that. Or maybe we thought some sort of mutation was going on. The rough strain mutated into the smooth strain and killed the mouse. That could have been a possibility. Or, and maybe you got this one, the rough strain ate the smooth strain bacteria and was able to be transformed into the smooth strain and kill the mouse. So a lot of different ideas, but hopefully as we go more through this experiment, it will give you closer to the idea of what actually happened. So hopefully some of you guys got that right. If not, that's okay. So leave your answers, don't change your answers, because that's your hypothesis. Okay, so experiment 5. Now they actually performed this experiment what they did in 4. The mice did get pneumonia and die in the experiment 4 that we look at right here. So the scientists propose a hypothesis to explain this. Somehow, in order for them to die from pneumonia, they have to have capsules. So somehow the pneumococcus cells without capsules gain the ability to produce capsules and can pass this ability along to their offspring. Remember that the capsule must be present for the cells to cause pneumonia. To test this hypothesis, the scientists prepared a mixture of nutrients in which the pneumococcus cells could grow outside the body of a living tissue. Such a mixture of nutrients is called a culture medium. The experimenters could grow the pneumococcus cells in a culture medium in petri dishes and observe them with microscopes to see whether the capsules were formed or not. The experimenters first grew large numbers of pneumococcus cells with capsules. They then killed these cells, ground them up, and dissolved them in a solution. Such a solution, called an extract, contained the materials ordinarily within the cell membrane. The extract from heat-killed cells with capsules was mixed with living cells without capsules. This mixture was added to a culture medium and allowed to grow. So according to the results of experiment 4, what is the most logical hypothesis as to the type of bacteria found growing on the culture medium of experiment 5? So the type of bacteria growing on the culture medium in experiment 5 would most likely be the smooth strain bacteria. Remember, smooth is going to be the one, if you look up here, smooth is the one with capsules. Okay, so it's going to be the smooth strain bacteria because that is the type that killed the mice. 5. Could these cells have produced... Oh, I skipped one. 4. Assume that the mice died of pneumonia in experiment 6. What conclusion... Oh yeah, it's up here. It says experiment 6 that some of the cells from experiment 5 were injected to healthy mice. So what do we think happened to the mice? What conclusion can be made about the cells? Again, the cells growing on the culture medium in experiment 5 were the smooth strain because it has to have capsules to kill the mice. So somehow those cells are gaining the ability to produce capsules. Could these cells have produced pneumonia in mice before they were grown on the special culture medium? And the answer is no, right? These cells without capsules, they cannot kill the mice with the pneumonia bacteria. So somehow between adding that heat-killed strain and how to gain the ability to develop capsules. 6. In the actual experiment like the one used in experiment 5 and 6, microscopic examinations showed the presence of live cells with capsules. So there we go. The live cells developed those capsules somehow. These cells could reproduce to form more bacteria with capsules. When these were injected into the mice, they caused pneumonia. How did the bacteria differ after they were grown on the special culture medium? Was the difference only in their structure? Explain. So a little hint there, not only were the original cells that were placed in the special medium changed, but their offspring also showed the same new characteristics. So something had to have been passed down from parents to offspring because the parents developed capsules and then their offspring did. So what could have happened? Well, the bacteria grown on the special culture medium were smooth and had capsules, giving them the ability to kill the mice. This change in their physical appearance, however, would be due to a change in their instructions. Hopefully we got this. DNA. Okay? So somehow their DNA changed to allow them to develop capsules. So let's look a little bit more into it, because if you think about us, our DNA just doesn't change. So how did this happen? So at the Rockefeller University in New York City, Avery, McLeod, and McCarty worked to find the part of the extract from the dead cells that caused the change in hereditary material. They wanted to find what it was that made it possible for cells without capsules to produce cells with capsules. They performed very careful experiments in which each part of the extract was tested. They hypothesized that some particular molecule in the extract was being taken up by the living cell, and that this molecule was responsible for the new hereditary information. The molecule was finally identified as DNA. Pneumococcus characteristics have been changed as a result of the incorporation of DNA molecules from the extract. So 7, what apparently happens to the DNA of the pneumococcus cells when they divide to form new cells? We kind of talked about this already. Once the parent cells got the new DNA and their DNA changed, they were able to pass that down to their offspring. And when they divide to form new cells, the offspring were able to produce these capsules as well. Alright, so if you didn't change anything so far, this is definitely what you want to have, because there could be test questions on this. So definitely make sure to have the answers for number 8 here, because this is the whole summary and what I wanted you guys to get from this experiment. So now what we know about DNA, give an explanation of what happened in experiment 4 to cause the mice to die from a pneumonia infection. Okay, and you can pause this too if you need to pause it to get it slower. So in experiment 4, the live rough strain, remember rough strain is the one without capsules, must have engulfed or ate the heat-killed smooth strain. So the heat killed the capsules around the smooth strain, leaving the DNA open, and the rough strain without capsules engulfed that DNA. This allowed the rough strand to take in the DNA of the smooth strain. The machinery of the bacteria was then able to read the old and new DNA, producing capsules on the rough strain, which made them smooth and gave them the ability to kill the mice. So that was the main thing I wanted you guys to get from this experiment. Okay? Once you are done getting this information, you guys are free to work on Activity 1. Remember, that's due tomorrow, Part A, just Part A. And I hope to see you guys tomorrow.